this is really as exciting as it can be for me. Many of you know Christy Ubert. She has been the driving force in the faculty research lunches that many of the faculty around Northwestern have attended. She has always gone well beyond the call of duty. She has been a pleasure to work with. She does so much for the NICO community and for people interested in data science and in collaboration. And she was sort of an obvious choice for being presented the Staff Service Award for NICO for all that she has done for us. And I would like you to join me in thanking her and with a round of applause for all her amazing contributions to our community. The second awardee is Thomas Stoger, who's receiving the Postdoctoral Fellow Service Award. And Thomas, just like Christy, has been outstanding. And I think so many of you that are graduate students or postdocs have interacted with Thomas on one of the many initiatives that he has kind of spearheaded to create new interaction modes and create new activities that enrich all of our lives at the university. And so it's really a tremendous pleasure and an honor for me to acknowledge him with this award. And so again, I would like you to join me in thanking Thomas for all his outstanding work. What I would also like to ask all the community is to think about nominating staff or postdoctoral fellows or graduate students that you believe have contributed to the enrichment of our experience here at Northwestern and at NICO. We are going to have these awards annually, and I think that to a great extent, the initial winners, both on the graduate student side and on the staff and postdoctoral fellow, were pretty obvious because of the magnitude of the contributions. But we want the community to actually tell us who they are noticing that are enriching all of our lives so we can recognize them. It's sad that we cannot recognize them in person, but at least we can recognize them virtually. So thank you, and we are going to be looking forward for your nominations for next year. So next, I would like to introduce Katherine Amato, who's a professor in the Department of Anthropology. And she kind of exemplifies what we believe is exciting about NICO and interdisciplinarity. Being someone that is very focused on the microbial side of things, which is not, you would think, a traditional area in anthropology, but actually addressing problems that have always been at the core of interests within anthropology, like health, hunger, and all of those things. So I think this is going to be an incredibly exciting talk, and I'm looking forward to it. And I will not spend any more time than what I already did, and let the time for Kathy to give her presentation. So let's welcome Kathy Amato to NICO. Kathy, take it on.
Thanks so much, Luis. It's great to be here. Let me just get my screen going here. All right. Can everybody see that okay?
Yes.
Yes.
Okay, there was a Zoom window open over it, so I wanted to make sure it wasn't making that weird shadow thing. Excellent. Okay, well, yeah, thank you again for having me. I'm really excited to be able to talk to everybody today. I've gotten a chance to share my research with people in some kind of smaller settings or in kind of different settings, and I'm really excited to be able to talk to this group on campus today. So as Luis mentioned, my work really lies at the intersection of anthropology, molecular biology, and microbiology. And so I want to give you a little bit of an idea kind of what that means today and some of the questions that we're thinking about in my lab. And this is meant to give you kind of a foundation for thinking about the questions we address and the methods we use to address them. But there's a whole lot more to build upon here. So I really will welcome people's questions. And I'm also open to talking with people about collaborating on things and thinking about kind of new perspectives we could bring in here. So to start with, the gut microbiome has become this huge area of research, the microbiome more generally. I focus on the gut microbiome. And one of the reasons it's so exciting is because it interacts with essentially any body system you can imagine. Kind of the three main categories I like to break it into, though, are nutrition, immune function, and behavior. So the gut microbiome breaks down compounds in the diet that are otherwise indigestible, like fiber. And gut microbes produce compounds like short chain fatty acids that can be used by hosts as an energy source that they wouldn't be able to get otherwise out of their diet without this microbial function. Microbes also can produce essential vitamins, they can break down toxins. And then some of these molecules that they're producing, like short chain fatty acids, are actually signaling molecules for other body systems, like metabolism. And so short chain fatty acids can actually affect how readily hosts deposit fat, or affect fasting glucose levels, et cetera. So there's a really interesting interaction in that kind of realm of nutrition and metabolism. We know that the gut microbiome also trains the immune function early in life. So germ-free mice that have no microbes of their own do not have a functional immune system, and you can rescue that effect by introducing microbes back in early in life. But if you wait kind of past a sensitive window, then you can no longer rescue that immune function. So there's a huge role there. And then we also know the immune system is constantly monitoring the microbiome, right? These are non-host cells that are in the body in close contact with host cells. And so the immune system also is shaping which microbes are there and how they're behaving as well. And then finally, we know that the microbiome can affect behavior. There's a whole lot of excitement about the gut-brain axis. And that the gut microbiome can actually produce neurotransmitters or precursors to neurotransmitters that can get to the bloodstream and pass the blood-brain barrier. The gut microbiome also can communicate with the nervous system through the vagus nerve, as well as through interactions with the immune system. And on the flip side, we know that things like stress and trauma can actually affect the composition of the microbiome and the types of compounds that it's producing. So these are really interesting bidirectional interactions. And because of that, it's perhaps not surprising that the gut microbiome has been associated with almost any disease you can think of. There are shifts in the gut microbiome that have been associated correlationally, if not causally, with diabetes, obesity, autism, asthma, Parkinson's, kind of you name it. Because of this, and because the microbiome is relatively flexible in response to the environment, right? Its composition can actually change over time in response to some of these variables like diet or infection status or stress. There's a lot of excitement about using the microbiome as a therapeutic agent, right? If we can actually get in there and change the microbiome, and then we know that it feeds back to affect all these physiological systems, it may actually be an area where we can kind of do a little bit more good than we can in kind of these more set areas, perhaps like host genetics, or even to some extent epigenetics. But because of this is an area that's been really highly investigated in terms of trying to understand what constitutes a healthy microbiome. And the field has generally come at this in kind of a biomedical way, right? In terms of kind of describing and assessing disease states and comparing those to healthy controls and trying to figure out what are the differences between disease and healthy states and what are similarities between different healthy states. And while this has given us some really good information, it's also left a lot of gaps. It's actually really difficult to come up with what constitutes a healthy gut microbiome. There aren't really clear patterns that people have come up with yet, except for a few kind of really general rules, like we think high diversity in the gut microbiome is a good thing. And there's a couple reasons for this. I think one, it's difficult to define what we mean by healthy, right? Do we mean healthy metabolically? Do we mean that in terms of, mental health? And kind of what do we mean by actually healthy? But I would also argue that there's a lot of different ways to be healthy, right? There's a diversity of cultures and lifestyles across the human population. And people live a lot of different lifestyles and have different cultural practices. And we know that these lifestyles and cultural practices affect the microbiome. So things like diet are going to be different from population to population, and diet affects the microbiome. And just because two people have a different diet and a different microbiome associated with that doesn't mean that one is healthier than the other. And so it's really difficult to nail down kind of what is healthy by simply looking at this kind of variation globally. And so one of the ways that my lab comes at this is thinking about it from an evolutionary perspective, right? We want to keep sampling diversity across modern human populations and trying to look for similarities between healthy individuals and differences between healthy and non-healthy. That's absolutely a valuable way to do this. But we like to think about it more evolutionarily. Because the microbiome can affect health, we think that microbes that are good for health, right, that affect metabolism positively, probably persisted across human evolution, right? And if you want to go that far, those types of microbes were probably selected for by the environment that was selecting for the phenotypes that they contributed to. And so you would expect those types of microbial traits to have persisted throughout human evolution. And there is some aspect to heritability of the microbiome in that parents often pass their microbes to their offspring, at least a portion of them. And so there is this potential for this kind of evolutionary dynamic to happen in which you could have the microbiome being selected upon and passed on across generations. And so we like to think about what it actually means to be human microbially in this kind of evolutionary sense, right? If we can figure out the types of microbes that have kind of persisted throughout human evolution, and that are possessed by populations today still, those are probably going to be the microbial either taxa or functions that should be considered kind of more typically human and healthy in terms of you need these types of microbes to be a healthy human being. So there's a few different ways to look at patterns in the evolution of the human microbiome and kind of how the microbiome has shifted across human evolution. The three main approaches are first looking at populations that share characteristics with some of our human ancestors, whether that be in terms of the diets they're consuming or the kinds of environments that they're living in. Another approach is to look at coprolites, right? So fossil poop, essentially, and see what do the microbiomes of ancestors actually look like. And a third way is to look at non-human primates, right? These are our closest living relatives. They have a lot of shared biological traits, but they have the upside that there's not as many kind of cultural and technological influences on their biology and on kind of the action of natural selection. And so by understanding patterns in non-human primates, both within primates as a model and understanding how different primates interact with their microbiomes differently in different environments, we can get some clues into what we think might be happening in terms of thinking about humans. Now there's positives and negatives to all of these approaches, right? Modern human populations are not analogs for ancient human populations. Coprolite records only go back a certain amount of time into the evolutionary record, right? And there's contamination issues with a sample that's been sitting out for thousands of years in the environment. And of course, non-human primates are not humans. But I think by kind of combining these approaches, we can really get an understanding of kind of what may have happened across human evolution. Now in my lab, we do a little bit of all of this, but we specialize primarily on non-human primates. And we do this in two ways. Like I mentioned, we look at non-human primates as models for understanding host microbe interactions and try to look at how microbes might be interacting with host physiology and ultimately affecting host ecology and evolution in these natural selective environments. And we also use non-human primates as kind of a comparative tool to understand how the microbiomes of humans might be different or similar to those of other primates, and again, give us insight into things that could either be unique or similar in the human microbiome compared to other non-human primates. We do this from a perspective of energetics and life history, predominantly in my lab. So just to give you an idea of what this looks like, we're thinking about how individuals extract energy and nutrients from the environment. We know that they extract energy and nutrients from the environment, and then they have to distribute that across different bodily processes, which we've categorized as maintenance, growth and reproduction, and activity here. And this has been something that's been studied for decades in anthropology and beyond. And often we think about how variation in host physiology and behavior can affect the different arrows in this model, right? Can behavior in terms of feeding behavior change the amount of energy that a host is getting out of the environment, or allow it to compensate for maybe increased needs, energy needs during periods of growth and reproduction, right, without affecting activity. And we now know that the microbiome interacts with host physiology and behavior and also has a direct impact on these processes. So as I said, the microbiome helps hosts extract extra energy from their diet and can also program metabolism and kind of help shunt energy and nutrients to different tissues. So we're really interested in thinking about this, how this changes over time or across populations within a species, and then again, comparing it across populations. So why does this matter when we're thinking about humans? Well, humans are unique among primates. We like to think of that as being humans, but it is actually objectively true in a few different ways. So first, we are the most widespread primate on the planet, right? Evolutionarily, we've been very successful, but as a result of that, that means that across our evolutionary history and in modern populations as well, we're exposed to marked environmental variation across time and space, right? So the environment is constantly changing and we're constantly needing to face different dietary challenges in terms of food availability, as well as things like changing infectious disease landscapes. And we know that the microbiome can have a role in mediating some of this. Humans also have expensive life history traits. So compared to other primates, we have relatively big brains, short birth intervals, and a really long lifespan, which means we invest a lot of energy over a long period, right? And it's possible that we're going through energetically demanding periods like reproduction and growth during periods of the year when there isn't as much food available, right? And we really have to deal with these nutritional challenges to an extent that other primates may not have to. Again, because we know the microbiome interacts with both diet and metabolism, it would make sense that the microbiome may play a role in contributing to human ability to support some of these energetically expensive traits. So this is really where we're coming at it and saying, in order to understand how the human microbiome has evolved over time and what might be healthy, what can we understand about the human microbiome in terms of its potential unique traits and how it differs compared to other primates? So that's the question that I really want to start with today, is thinking about, is the human microbiome unique? And can we link that to some of these other kind of traits that we know are unique in humans? As a first step towards this in the lab, we started by saying, can we just characterize the human microbiome compared to that of other non-human primates? So I amassed a data set of 18 species of wild primates. So these were all collected in the wild from across the primate phylogeny, all the way from lemurs to old world monkeys, new world monkeys, and then apes, and compared it to published data from 14 populations of humans. And these were humans that are living both in what we considered kind of industrialized populations, so eating kind of more processed foods, which are lower fiber, higher fat diets, which we know can affect the microbiome, and also more non-industrialized populations, so populations that are engaging more in hunting and gathering and are kind of less involved in this kind of industrialized food production chain. And we generated microbiome data we compared the microbiome data for these species and populations. And I'm going to skip over the methods a little bit, but I'm happy to answer questions if people are interested. But we basically use 16S amplicon sequencing, which allows us to say something about the taxonomic composition of the microbiome, and shotgun metagenomic sequencing, which lets us say something about the functional potential of the microbiome. So here's what the data look like overall. It's a lot to look at. First of all, these are ordination plots. Every point on a plot represents the microbiome of a single individual. The points that are closer together are more similar to each other, and points that are farther apart are more distinct. The axes don't mean a whole lot in and of themselves. And I've colored the top plot, I'll just say, is unweighted, so it's presence absence of microbial taxa, and the bottom plot is weighted by relative abundance. And then the points are colored by kind of general phylogenetic group. So New World monkeys are in red, lemurs are in purple, Old World monkeys are in blue, apes are in brown, and then humans are in orange and red. Humans that are colored in red are the industrialized human populations, and orange is non-industrialized. And what you can see really clearly here, first of all, which is an important point, is that they're grouping by phylogenetic group. Right? There's a lot of different dietary strategies represented among the primates here, a lot of different geographies, and all of that is kind of drowned out by this signal of host phylogeny. What was especially interesting to me, though, was this kind of bottom right-hand corner, particularly on this top figure that you could see. So you can see clearly that New World monkeys and lemurs are different from the rest. We also know that industrialized humans have a different microbiome from non-industrialized human populations. That's something that's been reported on before. But this bottom right-hand corner was really interesting to me. So I pulled out the New World monkeys and the lemurs and the industrialized human populations. And here I've just zoomed in on non-industrialized humans in orange, the Old World monkeys in teal, and the apes in brown. And what's really interesting here is that the human microbiome, as you can see visually on these plots, is more similar to that of these Old World monkeys. So these are things like baboons than it is to other apes. And that includes chimpanzees in that cluster, chimpanzees and gorillas. This holds up statistically, and you can also see it in the dendrograms on the bottom. In all cases, the similarity between humans and these Old World monkeys in terms of taxonomic composition of the microbiome is bigger, so there's more similarity between humans and baboons, for example, than there are between humans and chimpanzees. When we look at the functional data, the story becomes a little bit more complex, but kind of the punchline is the same. If you look at this plot in this dendrogram, you can see that it's a little less clear. And in this case, the humans are somewhat equally similar to apes and to Old World monkeys. But when we really dig down into some of the specific gene functions, which I'll talk about in a moment, you start to see those similarities coming back between humans and these Old World monkeys more so than humans and chimpanzees. We wanted to look at this in another different way. So we looked at kind of distinguishing taxa or pathways versus shared between humans and either apes or the Cercopithecines. And you can see in terms of the taxonomic composition, apes and Cercopithecines have kind of an equal number of distinguishing taxa when you compare them to humans. But Cercopithecines actually share twice as many taxa with humans than the apes do. In terms of the functional data, it's a kind of even stronger pattern where there are more functional pathways in the microbiome that distinguish humans from apes than distinguish humans from Cercopithecines. And there are slightly more pathways that are shared by Cercopithecines and humans than by apes and humans. And what's really interesting here, though, is that 26 of these pathways that are shared by humans and Cercopithecines are unique to humans and Cercopithecines. So the other apes do not have them, whereas everything that's shared by humans and apes is also shared by the Cercopithecines. And then when I was talking about digging in even deeper, if we go in and look at cazymes, which are carbohydrate active enzymes, these are the enzymes the microbiome uses to break down dietary carbohydrates. The strongest signal in the data set was that humans were most similar to baboons specifically in terms of these carbohydrate active enzymes. And that's compared to chimpanzees or any other primate species in the data set. So kind of the punchline here is that the human microbiome is most similar to that of Cercopithecines, more similar to that of Cercopithecines than apes. And this was surprising initially, right? Because chimpanzees are our most genetically closely related living relative. But it's maybe not surprising when we think about kind of evolutionary history and ecological niches. So extant apes tend to live in habitats that are usually rainforest habitats, and they have diets that are very much plant-based. Chimpanzees in particular are considered to be fruit specialists, and while they do include some meat and insects in their diet, really their physiology is set up for consuming fruit more so than anything. In contrast, Cercopithecines are living in more open environments and have a much more omnivorous diet that includes insects, small vertebrates, fruits, underground plant storage organs, right? So things like tubers, etc. And what we believe based on what we know about human evolution is that humans were inhabiting the same types of environments, or environments at least that were analogous to the environments that Cercopithecines like baboons are inhabiting today in general. And so we think perhaps that what's happening here is that the human microbiome actually breaks from the phylogenetic pattern that we see in all other primates, because our evolutionary history has broken from the pattern that we see in other non-human primates, and that our ecological niche and our physiology that matches that ecological niche is actually more similar to that of a baboon. I do want to mention, though, that there were similarities between humans and apes, right? It's not that there were no shared traits. There were absolutely shared traits and similarities. And so I also think it's interesting to think about the potential that the human microbiome could be a unique hybrid between that of an ape and a Cercopithecine. And perhaps the services provided by this kind of hybrid microbiome may have allowed us to survive as apes in these more Cercopithecine-like habitats. And so we're really interested in understanding kind of what are the traits that are more ape-like versus more Cercopithecine-like, and what sorts of impacts do they have on host physiology. The last two things I want to say about this data set that are also interesting, and again, thinking about how the human microbiome may have evolved and be unique or not compared to other non-human primates, is looking at variation in the human microbiome. There's actually more inter-individual variation in the human microbiome than there are in that of other non-human primates. So here you're seeing a plot kind of depicting the differences between individual samples, either from non-industrialized human populations or from the apes and the Cercopithecines put together as a group. And in the first plot, there's kind of the same amount of inter-individual variation in terms of taxonomic composition, which is interesting because, again, we have multiple species in that ape Cercopithecine group, whereas humans are a single species. So even the fact that there's similar variation is interesting and suggests there may be more variation in humans. And then in terms of the functional potential, there's actually more inter-individual variation between humans than there is between apes and Cercopithecines. And so again, it's really fascinating to think how we might get so much variation in the microbiome in a single species compared to across different species of closely related primates. And some of the hypotheses we have to do with things like dietary diversity, right? I mentioned how humans are the most widespread primate on the planet. And as we migrated in different environments across our evolutionary history, we had to adapt to a range of local environments. And so some of this variation in the human microbiome may be reflecting that kind of adaptive process and the role of the microbiome in it. And then the last thing I want to point out are some of the taxa that we are seeing as being kind of characteristic of the human microbiome. And so in terms of taxa, we have Helicobacter pylori and Bacteroides fragilis. And what's really interesting is that these are two key microbes that come up in a lot of the human microbiome health literature as key players in determining healthy versus diseased states. And these appear to be kind of characteristically human. In terms of the functional pathways, there are a lot of unknowns on this list, so it's a little hard to interpret the actual functional pathways themselves. But again, they've been associated with some interesting microbial taxa that again come up in a lot of the human microbiome literature as being potentially important in determining disease states versus non-disease states. So for this little kind of first foray into looking at what's going on in the human microbiome, we find that gut microbiome composition and function doesn't follow these expected host phylogenetic patterns, and that humans actually have a different microbiome than what we would expect if we looked at them compared to just only other apes, and they're actually converging with things like baboons. And it also looks like gut microbiome plasticity, or at least diversity among individuals, is increased in humans compared to other non-human primates. And so there's some hints in here that unique human ecology and evolution could be tightly associated with some unique microbial traits. So we've been thinking about ways to kind of go after this in more detail. And I'm going to talk about that again in terms of some of these kind of more observational and kind of pattern identifying studies that we've kind of built upon here. So kind of one of the next questions we had was, well, if there's so much inter-individual diversity in the human microbiome, is that related to diet? And can we find any patterns in terms of the human microbiome responding differently to changes in diet or differences in diet than that of closely related non-human primates? So to do this, we've started to amass a data set, and this is kind of in progress already. So I'll kind of talk you through some trends, but just be warned, there's more to do with this data set and more to add to it. But we got data from 3 species of wild primates. And for each of those wild primates, we had 2 populations. And real quick, these primates are from left to right, baboons, vervet monkeys, and then marmosets. So baboons and vervets are those Cercopithecines, the closely related primates that have very similar microbiomes to humans. And marmosets are a neotropical primate. So that's kind of an outgroup. For each of these species, we have species that are eating a human, or a population that's eating a human influenced diet. So in terms of the baboons, baboons that are raiding a garbage dump near a tourist lodge, compared to adjacent groups of baboons that don't have access to human food. The vervets, there's a population that's living on a farm and has access to crops and horse feed, compared to, again, an adjacent population that does not have access to those human food items. And then we have marmoset population. These are a little bit more geographically distinct, but one is living in the Caatinga environment, which is a very dry environment in Brazil. And the other is living in a forest environment, but outside an apartment building where they're often fed fruit and foods like that from humans. And then we have 4 populations of humans that I have in the data set right now, from Nuñoa, Peru, the Chimani in Bolivia, the Mosatan in Bolivia, and then Cebu in the Philippines. And these are kind of along a gradient of kind of industrialized food content. I do have data from people in the United States that I have to add to this to make the gradient stronger. But essentially the Chimani are hunter-gatherer group in Bolivia, and the Mosatan are a closely related group that has become more market integrated, and so they're consuming many more of these kind of industrialized processed foods. And then Nuñoa, Peru, and Cebu are, well, Nuñoa is a much more rural location as well, and there's been some kind of market integration happening over time, but the diet is largely reliant on kind of local agriculture still, and some incorporation of dairy more recently. And then Cebu is a large metropolitan area that has undergone kind of a transition to more of this kind of industrialized diet over time. So somewhat of a gradient here. And for this, we have the 16S amplicon sequencing, which tells us something about taxonomic composition of the microbiome. So the idea is, what happens when you have non-human primates eating this kind of more industrialized diet compared to humans that are eating a more or less industrialized diet. Here's what the ordination plot looks like. In green, you have your humans. And for all of these, the darker shade is the more industrialized diet, and the lighter shade is the less industrialized diet. In kind of your orange brown is the marmosets, the kind of yellows are the vervets, and the blues are the baboons. Again, you can see this is breaking up by species, right? So each species has a very distinct microbiome. You can also see the baboons are falling pretty closely to the humans again, but you can also see that as these primates are consuming these more industrialized diets, their microbiome is shifting towards that more of a human microbiome, right? Those points are closer to the human points than the points from the non-industrialized diet primates. When we look at the diversity of the microbiome, we have humans here on the left and then the non-human primates on the right. And in general, you see somewhat of a reduction in diversity with kind of a more industrialized diet in humans. And you see a much stronger pattern of that in the non-human primates, right? So the baboons, we have baboons that have full access to trash or partial access to trash, and they have a much higher microbial diversity than those that are, no access to trash and the partial have a higher diversity than full access to trash. And you have a kind of trend for a decrease in diversity for the vervets that are getting those human foods. What's interesting is the marmosets have an opposite pattern. They actually have increased diversity in response to human food intake, which is an interesting pattern. We also wanted to look at some of the taxa that we're shifting with these diet types. Firmicutes and Bacteroidetes are things that have been really important in human diet studies, and that we tend to see firmicutes going up with an industrialized diet and Bacteroidetes going down. In this data set, we're not seeing a huge amount of change in our human populations, but again, this may be because the diet gradient is not quite as extreme. I'll be interested to see what happens when I put the US onto this graph. But we do see, interestingly, a reduction in firmicutes and baboons and vervets to some extent, and then as well as the marmosets. So we're actually seeing kind of the opposite of what we might expect. And then in terms of Bacteroidetes, we actually see that going down as well in baboons, but then going up in the vervets and the marmosets. So we're not seeing the same patterns here necessarily. And then just the last example of taxa are Bacteroides and Prevotella. Again, these have come up traditionally in human diet microbiome studies, and that Bacteroides goes up, particularly with a diet that includes kind of more animal-based foods, and Prevotella goes down. Prevotella tends to be more associated with like a high starch diet. We do see to some extent the patterns we would expect in Bacteroides, in that they're higher in our kind of more industrialized diet human populations. They don't seem to change that much in most of the non-human primates, and non-human primates across the board have very little Bacteroides compared to humans. Although we do have the marmosets there with a couple individuals that have a lot of Bacteroides in that Western, in the human influenced diet population. And then again, in Prevotella, the patterns are kind of bouncing around here, and that, you know, in humans, there's not a clear pattern. In baboons, it is going down in response to this kind of human food, kind of lower fiber human food access. In vervets, it's going up, and in marmosets, it's not changing so much. So there's a lot to dig into here still, but some interesting patterns in terms of some things that are shared overall and some things that are different. And so, you know, so far at this stage in our analysis, this is kind of our conclusion, is that, you know, there are some shifts in non-human primates that are either different or shared with humans, but it's also interesting that the shifts we're seeing in the non-human primates seem to be of higher magnitude than what we're seeing across the human populations, which is kind of in contrast to what we were thinking about in terms of kind of dietary flexibility. But again, there's questions of kind of which species we're looking at in which populations. And I also do think we have species and or diet dependent patterns at work here, right? So these kind of human diets that these different primate species have access to are not the same, right? Getting access to garbage is not the same as eating horse feed, is not the same as necessarily getting fruit from an apartment building. And so we'd really like to kind of dig into the actual macronutrient content and what's happening in those diets more deeply. And then there's always outstanding questions, you know, does this hold with other human and non-human primate populations? Are these even the same microbes, right? We're looking at the genus level of microbe, but there's a lot of variation that could be included in that. And I think a lot of the microbial taxa that we're describing as the same genus in primates are actually probably very different microbes that are actually being found in humans. And then of course we have to look at the actual microbial function. So the last kind of little chunk of the story that I want to talk about today is thinking about whether some of these patterns in diet microbiome dynamics could be related to microbe fiber interactions, right? So when we think about industrialized diet, we're often thinking about a shift to a diet that tends to be lower in fiber compared to what a non-industrialized diet would be, whether we're talking about humans or non-human primates. We know that a reduction in fiber affects the microbiome based on human studies, and there's been some really cool mouse studies by the Sonnenberg lab also that have shown this sort of pattern, that taking fiber out of the diet will actually change the microbiome across generations in lab mice in a way that can't be rescued unless you introduce fiber and microbes back into the diet at the same time. And so we're certainly seeing fiber changes in these diet changes we were just looking at. But what's also interesting to think about, again, from a more anthropological perspective, is what about thinking about diet shifts across human evolution? We know that human evolution involved increased meat eating, domestication of plants through agriculture, cooking, and also potentially the use of fermented foods even farther back than the archaeological record. And all of these food processing techniques and food production techniques actually result in reduced fiber in the diet. They create kind of an easier to digest diet because there's less fiber. And this has actually been thought to be a key part of human evolution and what could have actually helped us be able to energetically handle the emergence of a larger relative brain size, for example. And so if humans have actually evolved to use a lower fiber diet, is the interaction between the human microbiome and fiber different from that of other primates just as at baseline? And are we kind of comparing apples to oranges in some ways? So again, to kind of start to get at this, we wanted to do a comparison in terms of fiber degradation between humans and non-human primates. Right now, and let me remind everybody that when fiber is degraded by the microbiome, these microbes produce short chain fatty acids, which can be used as an energy source. And so really what we want to look at is short chain fatty acid production genes. So this is a really good signature of understanding what the microbiome is doing in terms of fiber degradation for that host and in that microbial community. And so right now there's no good systematic comparative short chain fatty acid data that's actually looking at active production of short chain fatty acids by microbes from different primate species and humans. humans. So we're working on getting that together. But in the meantime, we decided to use a data mining approach. And this was led by my former postdoc, Dr. Liz Malott, who has since moved on. But what we did was we took existing shotgun metagenomic data, so the data that I actually showed you that we generated from non-human primates before, as well as available data from humans, and we wanted to look for these specific short-chain fatty acid production genes. We chose to use butyrate, a specific short-chain fatty acid, because we kind of know the most about this gene and it was the most tractable. Some of the other short-chain fatty acid production genes, either we don't know enough about, or there's like too many of them for it to even be realistic to kind of survey them in these data sets. So we updated an existing reference library of these butyrate genes, and then aligned our samples to protein hidden Markov models for each butyrate pathway gene using Xander. And then we aligned the metagenomes, our metagenomes to database to assess the prevalence and abundance of butyrate pathway genes. So we're basically mining these data for butyrate production genes. And what came out was interesting. So we expected potentially that there would be fewer genes in humans compared to other primates because we've kind of evolved toward this lower fiber diet. And what we saw to some extent was the opposite. So here on the left, you have humans, apes, catarrhines, platyrrhines, and lemurs. So catarrhines, again, are your old world monkeys, and platyrrhines are your new world monkeys, and the relative pathway abundances of these kind of different butyrate production pathways. And pretty much across the board, we saw that humans had higher relative abundances of these butyrate production pathways compared with all groups of non-human primates. And when we looked at humans only and split up industrialized versus non-industrialized, we actually did see what we would expect, and that there was a lower relative abundance of butyrate production pathways in industrialized human populations compared to non-industrialized. Although the difference was not that big, or was not as big as we might expect it to be. Another interesting kind of aspect of this analysis, and you don't need to like look at all of the bars on this slide, I know it's a lot to look at, was that in addition to looking at just relative abundance, we want to know which taxa were responsible for these butyrate production pathways. So we essentially looked for a complete butyrate production pathway and then would assign it to a specific taxon of microbe. And so these are the different pathways. Each kind of cluster of graphs is a different pathway, a different butyrate production pathway, or gene family, essentially, it is actually in here. And then each of the bars represents either humans, apes, catarrhines, or platyrrhines. And what you can see very clearly here is that there are different taxa that are more or less responsible for this trait in the microbiome, depending on which group of primates you're looking at, right? So not only do humans have a higher relative abundance, there's also different taxa that are providing this function in the microbiome in humans compared to other non-human primates. And so we think this is a really interesting area to explore further. These data suggest there could actually be increased potential for butyrate production rather than decreased production in humans. This is really interesting, again, because as I mentioned, short-chain fatty acids are an energy source. And so if you have increased production in humans, again, this could have been potentially contributing to energy demands as they increased across human evolution. Having more production of these short-chain fatty acids would probably have been beneficial for humans, despite reductions in fiber in the diet. And this idea that different taxa are performing these functions in humans compared to other primates, again, kind of hits home this idea that a certain genus in primate compared to that same genus in human may actually be doing different things. But either way, we think both of these patterns in the data signal an evolutionary shift in humans in terms of their microbiome and its relationship with diet that probably occurred as diet changed across human evolutionary time. Again, there's outstanding questions. So how ancient are these differences and have they changed over time? And then again, what about actual short chain fatty acid production, right? So humans may have more genes, but if we're eating low fiber diets, we may not actually be producing as many short chain fatty acids in practice as the microbiomes of other non-human primates. So we'd really like to keep kind of going after these data. So to wrap up, is the human microbiome unique? And do we think it's linked to our ecology and evolution? And can this tell us something about what's healthy in humans? And complement some of these biomedical approaches that are looking at disease states? Probably. And I hope I've convinced you that there's some evidence out there that this is very much likely the case. But as I've said, there's lots left to explore. So, in the lab, we really want to dig deeper into what makes the microbiome of different primates, especially humans, unique. We're really interested in determining which microbiome traits are most important for influencing host life history traits, right? Just because they're different between humans and other primates doesn't mean they're affecting host biology in a way that's meaningful, health-wise or evolutionarily. And we'd also, at some point, once we have a better handle on some of these interactions, like to better test the relevant contributions of microbial factors versus other factors to host life history, right? Microbiome's new and exciting and fun, but it's by no means the only thing that's, operating in this context. And so we have to remember the other factors that are playing a role and try to understand, is the microbiome more or less important or contributing, the same or different services. So I'll leave it at that. I'd like to thank my lab, which I don't really have a good picture of anymore, but I've kind of pasted a bunch together here, as well as a range of funding sources, the Department of Anthropology here, and our Evolutionary and Ecological Approaches to Health and Development group, as well as so many collaborators, co-authors, field assistants, and permitting agencies around the world that help us bring in these samples from so many different species and geographic locations. Thanks.
Thank you so much for an amazing talk. But I'm very upset with something that as you were talking, I was having these questions pop up, and then you proceed to answer them during the talk, which is very, very disappointing. But anyway, I'm going to go with it. James Gibb put in a couple of questions in the chat, and maybe you should read them, because otherwise I'm going to murder that Cerco-P blah, blah, blah word.
Yes. Cercopithecines. Yes. So could the similarity also be that due to geographic overlap between Cercopithecines, which has a wider geographic range than apes, and non-industrialized human populations, or the Cercopithecines crop raiding or exploiting human dietary waste? So absolutely. So that is one of our alternative hypotheses. I think I'll just talk about range overlap in terms of the kind of the raiding and exploiting human waste. The groups that we sampled for that particular piece of data I showed were not consuming large amounts of human dietary waste or crop raiding. I'm not going to say there was none of it, but there was very little compared to the next project I showed where we actually focused on that. There is potential that there's been this kind of transmission between species over evolutionary time, and that, these Cercopithecines are also widespread and have probably been in contact with human populations for a long, long time. And we know that microbes are spread both indirectly and directly through, social and physical contact. I think what would be really interesting to look at there, and we just don't have the data yet, this is why we're constantly collecting samples from different places and species, to look at, even within Cercopithecines, so within baboons, within macaques, there are certain species that live really well with humans, right? So things like rhesus macaques, right? We know that there are issues with in Asia in terms of they're totally fine living with humans. And there's other species that don't do as well. And so looking at those kind of finer scale differences in terms of, are the ones that actually live with humans and have overlap more similar than the ones that aren't, versus what diets they're consuming, I think would be a way to get at that. So it's a good question, and we can't totally rule it out yet.
So I had a question myself, and you sort of addressed part of it when you were talking about fermentation and cooking and stuff like that. And, and cooking in particular is very interesting to me, because I was thinking in terms of the effect of boiling something or cooking for a long time and how much that will affect the microbes that were versus eating raw foods, or versus fermentation, in which in principle you are not kind of destroying, but you are changing the distribution of some of the microbes. Is there a way to take into account the fraction of food that is sort of cooked versus eaten raw for some of these groups. I cannot resist hazelnuts and stuff, and there is a pleasure to it. And so I think that's in some group, and I think there is also a trend about eating raw stuff. So I don't know if that is something that is also taken into consideration and if it has a significant impact or not.
Yeah, so in theory, it can be taken into consideration when, with the primates, we're essentially following them around and writing down what they're eating. With humans, we rely on survey data, which has all sorts of caveats, right? Because people don't remember or don't want to tell you. But we can and do ask questions about that and can split it up like that. We haven't yet with these data sets. And some of the data sets we have, we need to build on and get better dietary data, if I'm being honest. But it does matter. I don't know if it's going to matter as much as like industrial, like fiber content, just straight up. But I will say Rachel Carmody at Harvard is doing some really cool work with cooking and the microbiome. And she's actually shown that she does work with mice. And she's shown that if you cook sweet potato versus meat and give it to mice, it changes the microbiome, but really only with the sweet potato if it's cooked versus not. The cooked meat versus the non-cooked meat doesn't have a huge microbiome effect. And there's also been some interesting studies starting to look at fermentation and how that can affect the microbiome. Because as you said, fermentation is a totally different process, breaking down fiber and toxins, which is why there are some foods we can only eat if they're fermented. But it also contains the microbes themselves, as well as short chain fatty acids that the microbes have produced, right? So it's a probiotic and a prebiotic. And I did some work on, I wrote a review on fermentation recently, which was fun because I hadn't thought about it a whole lot before. And we have genes to help facilitate consumption of fermented food and actual interaction with lactobacillus bacteria in our bodies that are like 10 million years old, which goes way back beyond the archaeological record. So I think fermented food consumption may have been really important in human evolution. But the last kind of fun anecdote I'll throw in there, not anecdote, fact, is that these Cercopithecine primates that we have really similar microbiomes to have cheek pouches that they actually hold food in. And a lot of the research on that has been associated with like food competition and social interactions, right? And be able to like shove a bunch of food in your mouth and run away. But they're actually highly enzymatically active. There was like a terrible experiment done in the 50s where they put like a piece of potato in a macaque cheek and like looked, forced it to eat it and looked at how long it took to break down. And it was like 50% of the potato was digested in like 5 minutes or something like that. So I think the Cercopithecines also have this kind of fermentation behavior that is done differently than in humans. But I think that's interesting to think about if that's some of that convergence also.
That is so cool. Can I ask a clarification question about the increasing diversity of the pathways for breaking fiber? And maybe I missed, were you arguing that because humans took in less fiber, that then they kind of diversify so they could take advantage of more of it, sort of be more efficient with the fiber that they were getting? And that's why it diversified. Sort of, if you already get in a lot of fiber, then you don't care if some of it goes to waste. If you are only getting a little bit, then it's better to get waste to process all of it. Was that kind of the idea?
I think that could be operating. I think that is one potential interpretation. I mean, I was surprised. I thought they were going to be less. When you do this experiment in mice, you take fiber out, it goes away, right?
Exactly.
So yeah, I think that could be operating. The other interesting thing, which I didn't say, is that some of those pathways, so the main pathway where you get butyrate being produced is fiber breakdown, but you can actually get butyrate being produced also through some pathways of protein breakdown. And so I also think there's been a shift in the human microbiome that it's producing some of these short chain fatty acids through different pathways that aren't coming directly from fiber necessarily. So it's probably a combination of both of those things. but I really want to generate the actual short-chain fatty acid data and play around with that. But we have to, get samples in unpreserved from far places, which is not trivial.
Yeah, I can imagine. It seems like you were so clear that people are not asking questions. You are able to ask questions on something else that was not addressed. So I'm going to give like 30 more seconds for someone to put a question in the Q&A box. Otherwise, I mean, if this was live, I would be actively shaming everyone and pointing at them for not asking questions. But this is
it's either so clear or so unclear. It could go either way. So.
No, it was clear. It was really, really fascinating. I mean, it's extraordinary, the finding, and there were lots of things that were, as you said, not intuitive, right? I mean, if I would have guessed, you asked me which one was similar, of course I would have said chimpanzees, right? I mean, it's like, and then it's not, and there is all this interesting stuff that is pointing out about, this evolution, right? The environment in which you were living had such a big impact, right? That overcame this huge difference in separation of the species. So it was really fascinating, and even I could understand a lot of it. So I imagine a lot more of the people. Well, I'm just going to shame everyone that is out there for not asking any questions, but I'm going to ask everybody to thank Katherine again for an outstanding talk. Really, really fascinating.